right. I don't have my remote with me, so I'm going to stand over here, but it's all right. So ostensibly what I'm going to talk about, and I'll get back to this in a minute, is this Genome Encyclopedia project that I helped coordinate at the Joint Genome Institute. The main reason I'm here is not directly because of that, but because I'm involved in this program funded by the Sloan Foundation to study the microbial ecology and microbiomes of organisms in the built environment, in buildings, cars, planes, et cetera. And we've noticed in our work in this area that there is a distinct lack of culture collections for the organisms from this environment. And we think that that's a really important future area for this field to be robust, as you've all given examples for all these other areas. So we'd like to learn from everybody here and, swindle, lobby, to, convince, con, all of you to somehow get interested in addition to the amazing collections that you're working on that largely relate to natural systems, that so-called unnatural systems might be really important to have culture collections from too. And Kyria also asked me to mention my lab is in the UC Davis Genome Center, and if people are interested in it, I'd be happy to talk to people about it. We have a pretty good facility there. It has 5 core facilities that anybody actually in the country can use if people are interested, although it's probably more heavily used at Davis. There's a metabolomics core, a proteomics core, a DNA technology core, which is largely sequencing, a X microarray core that we have now convinced to become a host microbe systems biology core, and a bioinformatics core. And I'm happy to tell people or talk to people in the breaks about the good and the bad and the ugly of how some of these things work. So I'm going to come back to the Genomic Encyclopedia project, which has been run at the DOE Joint Genome Institute, where I have an adjunct appointment. And I just thought I would tell you some of the history of this project, which started basically, I think this sort of, my involvement and some of the impetus behind this started before I was at UC Davis. I was at TIGER, the Institute for Genomic Research, sequencing genomes. And a couple of us who were more evolutionarily or ecologically opinionated on noticed this phenomenon that was highlighted in this great paper by Phil Hugenholz actually in 2002. Sorry if I'm in the middle here. I can just sit down on the ground. Which is Phil, we did the same thing. Phil took a phylogenetic tree of the bacteria at the time, highlighted the main phyla of bacteria, and then overlaid onto that tree where most of the genome sequences were coming from. And most of them at the time were coming from just 3 of the phyla, the Firmicutes, the Proteobacteria, and the Actinobacteria. And there was a smattering of genomes from some of the other phyla, but it was very poorly sampled. And as someone who was interested in a lot of things related to evolution and diversity of microorganisms, this limited range of the phylogenetic diversity being covered was a bad thing. And so we applied for and got a grant from the NSF from their Tree of Life program to sequence the first genome of 8 phyla, the only 8 phyla that did not have genomes available but included a cultured representative at the time. And so we started on that project, but I just thought I would point out and highlight this letter that we wrote in 2001 when we noticed a related problem. So Naomi Ward, who was at TIGER and had worked with Erika Stackelbrandt previously, had in particular noted that there were all these genome projects, and many of the genome sequences that were coming out were organisms that were just completely either incorrectly described or unavailable to anybody to work with. Only about 50% of the genomes, the organisms, were available in culture collections at the time. And this hurt us as people who, we didn't work at culture collections at the time, but we respected deeply what they were doing. So we wrote this letter and we proposed a policy which we have followed, not everybody follows since then, which is that the strains for which the genomes are being sequenced should be deposited in a culture collection so that they can be available to other people. We proposed, with no basis for picking the number, but we proposed at least 2 culture collections. It could have been 7 or 1, but we think that this is really important. We even wrote a sort of white paper on what we recommended people actually do, which was deposit their strains in the culture collection and then reobtain them from the culture collection prior to sequencing, so that whatever anybody else was obtaining from the culture collection was the same as what the genome sequence was from. And I speak from experience, we at Tiger were sort of pioneering microbial genome sequencing, and we sequenced the genomes of multiple organisms that were subsequently extinct. That is, we did not have the strain available anymore, or that we had the wrong name for, or that we said was the type strain when in fact it was not. And that created some hinky issues for us. Anyway, I just thought I would mention that, and I think that having a policy coming from a group like this and from culture collections for the community that is expanding massively the genome sequencing out there would be a really good outcome from some gathering of people. So anyway, we got these 8 organisms. Most of them we obtained from ATCC or DSMZ. We sequenced the genomes. We did lots of stuff. We thought we were completely brilliant. And yet it had virtually no impact on the areas that we wanted to impact. And that's because a single representative from 8 phyla of bacteria does virtually nothing to capture the diversity that is present in the bacteria. And so, there were, I forget how many there were, but there were something like 60 major lineages of actinobacteria. So even in the well-sampled phyla, there were very few the genomes were not sampled diversely within that group. So then we also noticed the same trend was true in the archaea and in the eukaryotes and even in the viruses. So the solution that we came up with was to really use the tree, not just phyla, as a guide in sequencing. So to fill in the tree, we really wanted to cover all of phylogenetic diversity, the 3 main branches in the tree of life. There were lots of plant and animal and fungal projects, but very few of anything else. So we proposed this genomic encyclopedia of microbes, GEM, in conjunction with the JGI, and the DOE and JGI people said that's really cool idea, but there's no way we're touching microbial eukaryotes. I mean, their genomes are enormous, they're messy, So we changed it to GEBA, the Genomic Encyclopedia of Bacteria and Archaea, and that's what launched this project. And in about 2007 or 2008, we started a pilot for the Genome Encyclopedia of Bacteria and Archaea project. It involved largely people from JGI or JGI affiliates. With the Department of Energy, this incredible machine was turned on to this phylogeny-based genome sequencing project. And I'll come back to some of the other participants in a minute, like the DSMZ, which we couldn't have done this without the DSMZ. And I just thought in my few minutes here, I would just go through a couple of the lessons that we learned from this Genome Encyclopedia project. Some of them are more about microbial diversity, and some of them relate directly to culture collections. So one lesson that we learned is that we selected organisms based upon their novelty in the ribosomal RNA tree of life. Now, it is true that the ribosomal RNA tree is not always perfect in inferring the phylogeny of organisms, but it's pretty darn good. And it turns out it was very good at predicting sort of the genome diversity of organisms as well. Again, the ribosomal RNA tree is not perfect, but the main point that we sold to the DOE was that when you get genomes from across the tree of life, it improves every analysis that you would want to do from across other organisms in that group. And I'm just going to show you one example of this. If you want to discover a new protein family diversity, and I can talk to people about this later, basically what we showed is that if you sequence a bunch of genomes from one clade or one family or one phylum or across the full diversity of life, the probability that you'll find a new protein family is much greater if you sequence phylogenetically novel organisms than if you sequence closely related organisms. Nikos Kirpides and his group showed that functional prediction of other genomes improves by having phylogenetic diversity represented. Nikos' group and our group showed that analysis of metagenomic data improved by having phylogenetic diversity covered. So there are lots of people now going into environments and sequencing the SNOD out of the environment in some way with metagenomics. If you don't have good, high-quality reference genomes from the phylogenetic diversity of the taxa in those environments, you're just going to cry. I mean, the analysis of that metagenomic data is going to be so much harder if you don't have those anchors in those groups. a thing that's really important that I admit I was blind to when we started this project was Nikos Kirpides and some of the other people involved really wanted to push this idea of having metadata about the organisms. And they partnered with George Garrity and colleagues and actually created the Standards in Genomic Sciences journal, which for every organism that's being sequenced as part of the Genomic Encyclopedia Project, they have been depositing metadata about that organism and writing a position paper, a little marker paper about the genome of that organism. Project management is really critical. And the last thing I want to say is another lesson that we learned deeply was the importance of culture collections for this project. So as sequencing has gotten cheaper and easier, even back then when we were doing this arcane 454 sequencing, ancient history, getting DNA was our rate-limiting step. Getting high-quality DNA that was not, impure was our rate-limiting step. And we went to the culture collections and said, we need help. We can't grow most of these organisms. We certainly can't get DNA out of most of these organisms. And DSMZ offered to do all of this for free. And so they not only grew organisms, they sent us these nice pulse field gel images of all the DNA that they isolated. They quantified it, they characterized it, and they kept some of it so that once we finish the genome, people can order the DNA from DSMZ. So I think I'll just really quickly skim through this. We convinced people that this was worth doing. There's sort of a GIBA cyanobacterial project, a GIBA halophile project, a GIBA root nodulating bacterial project. And in the future, I just want to say one thing in my last minute here, which is that culture collections are grand. I love culture collections. They are fundamentally important to everything we were doing here. However, most of the diversity of microorganisms is not yet represented in culture collections. So there's been a spin-off from this project, which was run by Tanya Wojcicki at the DOE JGI called the Microbial Dark Matter Project, which has been trying to generate genome sequences from lineages for which there are no culture representatives, but which are phylogenetically novel. And interfacing that work with culture collections is still, I think, important. So not everybody is vouchering environmental samples. I think that's one thing that culture collections could do. And I'll just end by mentioning that it just got announced yesterday that we're part of a new grant run by PI as Ramunas Stepanovskis at the Bigelow Labs on doing this microbial dark matter project to try and go through the novelty in the tree and really generate genomic diversity. And I will just, so Kyria doesn't kick me out of here, I will just end there. Yeah.
What do you do to get a sequence your first collection organism and there's 100% identity for something in the database from uncultured clone?
Uncultured clone, is that what some people label it as? Or, yeah, I mean,
Uncultured
if
soil
the
fungus.
method cleans up and you get a lot of sequences of uncultured organisms, but you have a collection over your hand and it says 100% genome identity.
I don't know this. I mean, it has to be fixed, but yeah. Yeah. All right.
For our last presentation for this morning session, Vice President Mukherjee is going to be information on the EVA project, and it's called the Genomic Exploration of 1,000 Microbial Collections. Okay, thank you for the invitation. I understand that it's difficult to follow up a 100,000 genome project with only 1,000 genomes. But thanks, Jonathan, for the introduction to GIBA. As Jonathan mentioned, the GIBA project was followed up with several other pilot projects like the GIBA root bacteria, the GIBA cyanobacteria, the GIBA single cell microbial dark matter project, and the GIBA KNG or the 1,000 type-string projects, which I'm going to be talking about today. So for the GIBA KNG project, 1,000 phylogenetically diverse microbes were sampled from all over the world. Most of the DNA and isolates came from these two type cultures, namely the DSMZ, where we got most of our DNA from, and American Type Culture Collection. And in total, we have around 1,005 phylogenetically distinct type strains in this collection. So they represent 21 different phyla
it includes the top 4 phyla, which are well studied, like the bacteria, proteins, but we also have a lot of other phyla which are not as well studied, like the Galactrixie, where we have one sequence representative, then we have different bacteria, some of these bacteria, and so on. So not only do the cane genomes come from different parts of the world, they come from a wide variety of habitats, starting from Mediterranean Saloon Lake in Spain to soil from South Korea to the human microbiome or the toad of the line. So there's a lot of diversity with respect to habitats as well. So talking about diversity, I thought I'd take this quick detour to say how JGI is sequencing a wide variety, wide diversity of microbes. So if you look at the bar chart on the left, this is a number of isolated genome sequencing projects by Sequencing Center. And as you can see, the broad leads by the total number of sequencing projects followed by JGI. But when you look at the unique genus and species that is being sequenced, JGI is well ahead of the others. So talking about diversity, I wanted to see how much diversity we are adding with the KMG genomes in all the phyla. I was specifically interested in the well-characterized phyla, for which we have many sequence representatives. So for this exercise, what we did was we took all the sequence genomes and looked within each particular phylum and looked at the cumulative increase of 16S distance within these phyla. Then we added the KMG genomes to see how much that increase occurred with KMG with respect to random isolates of non-KMG genomes. And as you can see, even for a phylum which has thousands of genome representatives, like bacteria, we see nearly a 2-fold increase with KMG genomes as opposed to random isolates of other bacteria. And this number goes higher for cuties, actually bacteria, and we see the highest 5-fold increase for fusobacteria. So to give you a brief overview of the analysis that we have planned and that is underway for this project, the cornerstone of this project, the main goal for this project is the metagenome binning, as Jonathan was mentioned. So we are looking to see how the KMG genomes are improving taxonomic assignment in metagenome samples. So this computation of nearly 2,500 metagenomes with all the isolate genomes completed this past week, and it was done for a total of 6 million computing hours. And so this is our main focus for this project, and I'm going to show some preliminary results for this. And then we are also looking at a distribution of CRISPR-Cas genes. So they are listed on this block, very hot topic right now. So we are looking at how CRISPR-Cas systems are distributed across the KMG genomes, and we're comparing that with non-KMG genomes as well. And we are looking to see if we can identify new CRISPR-Cas